So we're going to move on to our next speaker, Hongyu Chen, who's a postdoc researcher at Zhejiang University in China. Unfortunately, he could not make this live session today because of time zones, but he sent us a recording and he also put his email in the slide if you want to reach out to him via email. So today he's going to talk about identification and annotation of cell clusters using plant single-cell RNA-seq database.
Hello, everyone. I'm incredibly honored to be a part of this presentation and to share my insights with all of you. I regret to inform you that due to the unavoidable circumstances, such as time zone differences, I'm presenting this in a recorded format, and I hope that the content I provide today will still be vulnerable and insightful for you. Please feel free to reach out to me through email, and I will do my best to address any question and the discussions. So today my presentation topic is PlantscRNADB, how to fascinating single-cell research in plants. I will mainly introduce the plant single-cell database we previously constructed, which is now updated to its third edition. Additionally, I will provide an overview of the current work we are conducting. The progresses in plant-based research may not match the repeat pace observed in animal studies. The field of single-cell application within the domain of plants is witnessed a notable acceleration. As of April 2023, the publication count has exceeded a significant milestone of 100 articles, attesting to the burgeoning interest and adoption of single-cell methodologies in plant biology. Despite this burgeoning interest, a critical challenge remains: the effective exploration and utilization of this wealth of data. Presently, the accessibility and user-friendliness of databases dedicated to plant single-cell data exploration are in need of significant enhancement. Creating and refining intuitive platforms that cater specifically to the complexity of plant-based single-cell database will undoubtedly facilitate deeper insights and propel discoveries within the field. Improving the interface between research and data is crucial. Enhanced database functionality, more robust search article algorithms, and intuitive visualization tools will enable scientists to navigate, analyze, and interpret this expanding repository of plant single-cell information more effectively. By fostering this advancement in data accessibility and exploration, we can unlock a wealth of knowledge that lies within the world of plant single-cell research, fostering innovation and driving the field towards new horizons. Using single-cell transcriptomic data has unlocked numerous pathways for analysis, allowing us to exploring various aspects of cell biology, from cell clustering to understanding cellular communication, tracking cell trajectory, and delivering into RNA velocity analysis. These analytical techniques provide valuable insights into the complexity of plant cell dynamics. However, amidst this advanced analysis, one fundamental and critical aspect stands out: cell annotation. You might wonder, why is cell annotation so crucial? Well, let me break it down for you. Simply put, cell annotation involves identifying and characterizing different cell types within a sample. is likely giving each cell an ID card, telling us what it is and what it does. This step is important because without accurate cell annotation, all other analyses could be hindered, even leading to inconclusive results. For instance, take a look at this cell clustering diagram on the left. It's from our single-cell transcriptomic sequence of tobacco leaves, resulting in 21 distinct cell clusters. Without cell type annotation, our analysis could seem quite perplexing. However, by annotating each cell type using homologous genes from other plants and specific marker genes from tobacco, we can define each cell type. For example, cluster 11 and 17 are significant set for aroma synthesis in tobacco, known as groundry trichomes. Therefore, by accurately annotating cells, we gain a deeper understanding of specific cell types within the sample. The origin lies the foundation for further analysis, allowing us to discern unique functionality and the role of different cell populations. Cell annotation provides the background needed to unravel the complex biology process within plant cells. Why cell annotation is a hurdle in plant single-cell analysis? Well, let's take a look at the current mainstream approaches to annotation. Looking at the diagram, when it comes to annotating a new single-cell transcriptomic dataset, we can approach it from three angles. Firstly, one way is to leverage a well-annotated reference dataset. We match the new data with this reference set, assessing exploration similarity to determine cell types. Yet updating a well-understood reference dataset itself poses a major challenge, creating what seems like a circular dilemma. Secondly, another approach involves the cell type specific marker genes. Ideally, this gene is expressed only in a particular cell type and not in others. If a specific group expresses this marker genes, it's defined as that cell type. However, currently such marker genes resources are crucial in the realm of plant studies. Lastly, validating cell annotation results based on cell function is the key. Specific cell type showcases distinct functions. For instance, in our previously analysis of tobacco leaf cells, after annotation, we conducted geo-enrichment analysis on the cell groups. It revealed fascinating insights. Leaf mesophily cells exhibited enriched in photosynthesis or carbon fixation pathways, while impermeable cells enriched in pathways related to fatty acid metabolism or wax synthesis. This finding aligns with peer research. Thus, due to the security of reference datasets and cell type specific marker genes in plant domain, annotating plant single cells currently present certain difficulties. So we are building a database for plant single cell transcriptomics to track those two issues. One, I mentioned earlier, when making it easier to exploring the plant single cell transcriptomic datasets already published. 2. Giving you a bunch of marker genes to help analyze new plant single-cell transcriptomic. Currently, the database has been updated to its third version, encompassing 15 plant species and featuring a workshop 110,000 mark genes. In this database, mark genes primarily stem from 4 sources. Firstly, there are genes validated through experiments show specific expression in certain cell types. These are the most reliable but are relatively few in number. Secondly, we've identified highly expressed genes in specific cell type through background RNA-seq data analysis, yielding a higher count due to the differential expression analysis compared to experimental validated source. However, the majority of marker genes came from single-cell data analysis. Since different studies use varied criteria to identify cell type specifically expressed genes, so we've reanalyzed each dataset using uniform standards to pinpoint different differential genes, considering them as a form of the evidence. Looking at the number of tissue and cell types, it's clear that current plant single-cell research predominantly centers around model plants like Arabidopsis and rice. However, it's currently broadening its scope to include a wider range of species. Adobe Process takes the lead with 22 tissue types and 275 cell types. It is kind of peculiar why are there so many tissue types. They point towards focusing on the cellular hierarchy structure within our database. First off, in order to replicate the initial findings from the articles, we adopted a simple two-tiered structure while retaining as much detailed information as possible. For instance, although lateral root logically falls under the root tissue types, and saturatin should belong to the leaf tissue types. Presently, we've displayed all the detailed information. Likewise, the century applies to cell types as well. Besides the data overview, the current database offers various search functionalities categorized into 2 types. The first tab enables users to trace from cell types to their corresponding marker genes, while the second tab allows users to examine genes and identify in which cell type they are expressed. To navigate from cell type to their respective marker genes, users need to go through a series of selection. Start from species, then tissue, and finally cell type. Once selected, the database provides the marker genes associated with that specific cell type. We've conducted an intentional scrambling of these marker genes. If a gene exhibited over 80% expression slowly with that particular cell type in individual expression data, we defined it as marker 1, while others fall under marker 2. Additionally, the search results also include corresponding data source information using NCBI project IDs as an example. Users can leverage these marker genes for annotating and analyzing newly generated data. The gene-to-cell type search function offers an alternative approach to exploring plant single-cell data. By selecting a species and importing the corresponding gene names, user can discover which cell type and identify these genes as a marker, along with evidence from which articles supporting this identification. Additionally, we present UMAP diagram offering a more intuitive visualization of how the gene expression across various data sets. Though single-cell genomics has seen extensive application in the realm of plants, it lacks information about the spatial presentation of cells. Spatial transcriptomics technique aim to address this gap. In the third adaptation of our database, we've included published spatial data from the plant domain, encompassing 4 species. User can now precisely pinpoint the specific spatial location of gene expression. So through this function, we can now to some extent solve the 2 different program in analyze plant single-cell data mentioned earlier. However, as we access our database, we've delved into identify its shortcomings. We've currently refined 4 key aspects. Firstly, our focus is on standardizing cell types. Presently, the database lacks a well-organized and uniform system for tissue and cell types. It's a bit convoluted as it aims to mirror the original articles lacking user-friendliness. Different articles may describe the cell type differently, lacking a uniform and systematic approach to defining and organizing these cell types. Hence, we are looking to realize the concept of plant cell ontology. For instance, considering life structure as shown in the diagram below, we aim to compare a standardized hierarchy structure of leaf tissues and cell type by reference database like plant ontology or bar database. For instance, leaves should be categorized into epidermis, vesiculture, and mesophyle, with epidermis further divided into stroma, trichome, and pavement cells. This structure will serve as the detailed reference for annotating cell types in plant single-cell transcriptomic data analysis. Currently, our efforts are focusing on standardizing the cell type and structures of respective specifics like rice and adobo opsis. The functional features of cells reflected the biological activities and characteristics specific to certain cell types, which may exhibit similarity across different plant species. For instance, pair studies have revealed functional similarity in cell types within the same tissues across different species. For example, both Arabidopsis seedlings and tobacco leaves are shown in the diagram, exhibited noticeable enrichment in photosynthesis-related geopathways within their mesophilian tissues. Therefore, by understanding and analyzing cell functional models or pathways, we could potentially improve the identification and annotation of various cell types. This understanding made very similar or related biological traits even across different plant species. These functional models as annotation tools shifted the focus from individual genes to the functional information derived from multiple genes. This shift aids in uncovering differences between cell types, fostering a deeper, comprehensive plant cell function and characteristics. Lastly, besides protein coding genes serve as marker genes, recent findings demonstrated that single-cell sequencing can also detect a significant amount of information regarding non-coding RNA expression. Plant genomes generate abundant non-coding RNAs, typically expressed in specific environments or tissues, playing important roles in regulating various biological processes. Exciting research as shown in the diagram. Similarity indicates that non-coding RNAs like protein coding genes can serve as a marker for cell types. Therefore, updating and focusing on non-coding RNAs will be a priority for upcoming data reversions. This far, PlantscRNADB provides an excellent platform for exploring published plant single-cell data, offering marker genes that aid in annotating new data. So the upcoming updates in 4 areas will enhance the accuracy of PlantscRNADB, providing greater convenience and further insights. So thank you everyone for your attention. This concludes my presentation.
Awesome. Awesome. Thank you so much to Hongyu for that. And like I said, again, if you guys have any questions for him, feel free to use that email that has been put in the chat, and you can send him an email, and I'm pretty sure that he'll get back to you.